Hi everyone, we will do the revision of abnormal hemoglobins and sickle cell anemia. First question is abnormal hemoglobins. Hemoglobin has 4 polypeptide chains and HbA is the major hemoglobin found in the RBC of adults. It has 2 identical alpha chains and 2 identical beta chains. Alterations in the amino acid sequence of polypeptide chain can produce altered conformation and abnormal function and disease. Example for abnormal hemoglobins are HbS, HbE, HbC, HbD, HbM, etc. So, HbE is the second most prevalent variant. So, here glutamic acid in the 26th position of beta chain, it is replaced by lysine. It has same mobility as of HbA2 on electrophoresis. In normal hemoglobin, 6th amino acid in beta chain is glutamic acid, but in HbC it is replaced by lysine. Heterozygotes, they have moderate disease and homozygotes will have hemolytic anemia. Next one is HbD. HbD, it results from replacement of beta 121 glutamic acid by glutamine. It does not produce any sickling. HbM are a group of variants where the substitution occurs in the proximal or distal histidine residues of alpha or beta chain. So, as a result, the heme will have a tendency to get oxidized to hemine, forming methemoglobin. So, here the oxygen binding capacity is decreased, which will result in cyanosis. So, the molecular differences in HbA, HbS and HbC are: in HbA, 6th position of beta chain is glutamic acid. In HbS, this is replaced by valine, and in HbC, it is replaced by lysine. So, in HbS, at 6th position of beta chain, glutamic acid, it is replaced by valine. HbC, glutamic acid is replaced by lysine. HbE, 26th position, glutamic acid is replaced by lysine, and in HbD, glutamic acid is replaced by glutamine. Second question is sickle cell hemoglobin or HbS. It is the most common variety. Here hemoglobin with abnormal electrophoretic mobility is responsible for the sickling disease. The glutamic acid in the 6th position of beta chain of HbA is changed to valine in HbS. So, this will lead to the polymerization of hemoglobin molecule inside the RBC. So, the substitution of hydrophilic glutamic acid by hydrophobic valine, it will cause a localized stickiness on the surface of the molecule. So, that will cause distortion of the cell leading to the sickle. The deoxygenated HbS, it has a protrusion on one side and a cavity on the other side, so that many molecules can combine and polymerize. HbA and HbF, they do not co-polymerize with HbS, so they will prevent sickling. HbS can bind and transport oxygen, and this sickling, it occurs in the deoxygenated state. The sickled cells, they will form small plugs in the capillaries that will lead to occlusion of the major vessels, which can lead to the infarction in organs like spleen, and death, it usually occurs in the second decade of life. Electrophoresis at alkaline pH shows a slow moving band than HbA. At pH 8.6, carboxyl group of glutamic acid is negatively charged. As we already said, in sickle cell hemoglobin, the glutamic acid, it is replaced by valine. So, the lack of this charge on HbS makes it less negatively charged and decrease the electrophoretic mobility towards the positive pole. This is in the alkaline condition. But at acidic pH, HbS, it will move faster than the HbA. Next is sickling test. So, here a blood smear is prepared, then a reducing agent such as sodium dithionate is added, then the blood smear is examined under the microscope. So, that will show sickled RBC. In sickle cell trait, 50% of the hemoglobin in the RBC is normal. So, this sickle cell trait as such it will not produce any clinical symptoms and the person can have a normal lifespan. But at high altitudes hypoxia may cause, then chronic lung disorders also produce hypoxia induced sickling in HbS trait. Management of sickle cell. Repeated blood transfusion may be required to treat severe anemia, but this can also lead to iron overload and cirrhosis. Then treatment with anti-sickling agents like urea, cyanide and aspirin, they will interfere with the polymerization.